start the class now. Sundus, if you can share the chapter 13, coordination and response. I'll give a brief introduction, and you start recording in the local computer so that we can upload it later on in the YouTube.
Okay. Let me turn on my video.
Mm-hmm.
Okay, so this is the first page of the chapter.
Oh, yeah, that's good. So is it visible to everyone? I hope so. Enlarge it. Yeah, this is fine, inshallah. So today, inshallah, we'll talk about this coordination and response. I think if you remember what exactly is coordination and what is response, when we go through this chapter, you will understand what do you mean by these terminologies, and they are really very important. But this is basically dealing with the central nervous system, how your brain or your central nervous system is coordinating activities going on in your body. So it's not talking about the reflex arc or reflex action, but we have many activities going on each time: your blood pressure, your heart rate, kidney filtration, for example, thinking, thought process, recalling something, your eye reflexes, and sense, smell, light reflexes, and many, they are very much coordinated. And how these activities are coordinated by the central nervous system, inshallah, we will have a scientific discussion. And as you can see here, the objectives are very clear. You could see in the first box in this chapter what exactly we're going to cover. So first of all, human nervous system, because we are more familiar with our system. So I think this is really a very good discussion to understand how central nervous system works. And then you have neurons. As we mentioned about the renal system, if you remember, nephron was the functional unit, that was the main player. So here in the central nervous system, you have neurons. If you understand the function structure of neurons and how do they communicate with each other and how they are regulating different body functions, I think that will make things very easier. So there's wide types of neurons and type of the what kind of communication is taking place among neurons, that we will talk about. Then we have reflex action. This is one example actually to understand that how this body is regulating, central nervous system is regulating or coordinating different functions. And then you have structure and function of the eye is another example, which is a very best coordination because this is again very common. Your eyes blinking and in the formation and lubrication of the eye when you are blinking it, and how it is responding, the pupil size of the eye. If you put torchlight in your eye, how it constrict the pupil size so that the retina is not damaged. And when you expose all of a sudden coming out of your house in a bright sun, you could see that your eye will immediately respond to that, and that will decrease the intensity, that will decrease the pupil size, the entry of the light, so that your retina is not damaged. So these are, and then you have hormonal system, which is again, so you have two major types of coordination system. One is through the central nervous system, your brain and spinal cord, and another is through hormonal system, the glands that are secreting different hormones like adrenaline. When you start running or when you jump, for example, or fight and flight reaction. If you, for example, somebody will see snake or somebody will see lion, for example, all of a sudden frightened response, immediately start running. So the heart has to race, it has to increase the contractility so that it can easily pump more blood, and it prepares your muscle and your respiratory system and all the energy system so that you can respond easily. So here in this chapter, the trigger is reaction times. So they have given us a time of sprint. So I would like to know from you, as you know that in these sessions, we would like to have a interactive discussion. So it's not just covering the curriculum, but I want to challenge your mind. I want to challenge, we should challenge each other to see how we analyze these information, right? If you can understand the basic concept and the basic mechanism of these different description in the chapter, I'm sure that it will consolidate your knowledge and you can recall it in the exam as well, and you can recall it anytime when you want to use that knowledge. So let's talk about sprint. They have mentioned reaction time. Sprint in this example is given in coordination and response chapter. Who would like to tell me what comes to your mind why they would think about sprint? In the chapter of coordination is a trigger. As you know, the sprint is one of the most popular activities, sprint races, for example. So anyone would like, you can raise your hand and let me know, please. Okay, is there someone to talk about sprint? What exactly, why they use this trigger and what is mentioned here? We will have a brief summary, inshallah, of this trigger, and then Sundus will take over. Okay, we could see Zainab, Afan is here. Okay, could you give chance to Zainab to respond, please? And then Kashif, you can also add. Yes, Zainab.
A sprint is like a race in which all of the people have to run around, and at the starting of the race they shoot a gun or something, but
Okay.
since sound takes time to travel, so they have got other mechanisms by which all of the runners know at the same time that this race has started now, and so they have a reaction time for it, and that's why they mentioned it in this chapter over here.
So this reaction time is how this reaction time is controlled.
The reaction time is the time is like how much time your body takes to react to a certain stimuli, and in this case, the person who is nearest to the gun being fired, he would hear the sound quicker than the person who is furthest away from the gunfire. So since he's the furthest away, the sound would take the most time to reach him, and he would react the slowest. So to make it fair for everybody, they use different mechanisms, so that all of their reaction times may start from the same time.
Yes, that's great. Wonderful. Yep. Yeah, this is a very beautiful explanation, especially she mentioned about the stimulus. So in the core knowledge of this chapter, you need to understand what exactly stimuli or stimulus, what is receptors, what are effectors, for example, what are neurons. These are core knowledge. If you don't know about these different terminologies and their function, so definitely you won't be able to grab the major concepts. Yeah, excellent, Zainab. Afan, you would like to add something?
No, sir.
Okay, is you're happy with the explanation? Okay, great. So here they mentioned the reaction time. Actually, as she mentioned that if you're starting the race with a gun sound, so the person who is standing near the starter definitely is going to hear the sound very quickly, and he could respond immediately. So as you know, in sprint, the pickup time, the moment you respond is extremely important. With a split of second, with the difference of split of second, a person may lose or win the race. So they have another mechanism, as she mentioned, the stimulus, that electricity or light stimulus, for example. So they could look at that stimulus immediately. So when the signal is on and there is a sign of light, they start immediately. So picking up, actually, that is the response, which is very immediate, and that is very quick. If you go through the whole description, they have mentioned that this is, for example, who won the race in the 100-meter final in 2012, for example, the third paragraph. So they have given also the timing, like how they have responded. For example, Johan had a reaction time of 0.17, and there was another 0.178 seconds. So look at 0.165, and the difference 0.179 is not a big difference, right? But he won gold medal. Why? Because he reacted immediately. And then they have a standard time as well. Some people are really reacting immediately before even the sound, or their standard reaction time is very low. So they have a cutoff time that you need to respond in this particular time. It should not be less than that. It could be immediately, or depending how your body is prepared and how your system is coordinating with each other, and that will help you in winning the race. So I think, yeah, this is a trigger because this is strictly coordinated by the central nervous system. If you exactly remember the nerve, which is passing the longest nerve fiber, the longest neuron in your body, from the lower part of the spinal cord, going directly to controlling the toe, right? When somebody is running, you could see in the picture, so why they are standing on their toes rather than heels? Which one is responding more? Is there any explanation for that? That it's better that you have a little bit incline on your heel, right? Your heels are up, they are not touching the ground. Your toes are strictly touching and the ground, and that will help you for the pickup. Immediately you could pick up because your nerve can respond very quickly. We have this discussion actually regarding this picture. The neuron which causes sciatica nerve, for example, is the longest one going through, and that is controlling the movement of the toe. So before discuss further mechanistic in this reaction time, let's talk about how our brain is working, how our central nervous system is controlling our body system. So over to Sundus Bukhari to explain, and we will be with you inshallah if there is any gap.
Okay, thank you. So this is I mean, in on this page, they are discussing the difference between reaction time and reflexes. So it's very important to know that these two things are not the same thing. Okay, reflex is different from a reaction. So reflexes are involuntary, and it's something that the body does itself, and it is used to protect the body and are way faster than a reaction. So an example of a reflex is, if you go to the page, 164 in your book, figure 13.7 over here. The tapping of the knee and then this leg jerking upwards is an example of a reflex. So this is the protection of that tendon. So the body is straightening it out so that it doesn't break or is not stressed. Whereas reaction is something, is the response to a stimulus. Okay, so the reaction time is a measure of the quickness with which an organism responds to some sort of stimulus. So I know there are lots of terms that you don't understand over here, so I'll clear that up in just a second. So the normal reaction time is different for the visual stimulus and auditory stimulus and then the touch sense. So that's because of the difference in which in the way these particles travel. And you know, light travels faster than sound, and so there's also like physics and chemistry over here. Okay, so we're gonna just take them all together. So so coordination, there are two types of coordination, types of way our body like communicates with different parts, different ways in which the brain communicates with different organs of our body. There are two systems. One is the nervous system and the other is endocrine system. And so the basic units of both of these systems is, for the nervous system, neurons, for the endocrine system, hormones. So we're going to be focusing on the nervous system for now. So for now, I'll explain what all those terms meant. So a stimulus is a way the effector in our body interprets certain change in the environment or inside the body. So let me give you an example. For example, someone places their hand on a hot stove. So now the stimulus is the high temperature, and the receptor is the receptor cells on our skin. Okay, and then the so the receptors then send impulses up our neurons, and I'll tell you how in just a second. And then when we get the message from the brain, uh, it's going to send the signals to the effector organ. So over here, the effector organ will be the muscles in the hand, and that's how the person is going to remove the hand from the stove. Similarly, there can be an internal stimulus. So an internal stimulus, an example of that is, for example, um, low amount of water. So the brain detects the low water. Okay, so it has receptors. There are receptors in the brain, and then it sends, so the receptors send electrical impulses up the neurons to the brain. Then the brain sends impulses to the effector. Then that effector will be our thirst glands. So our thirst. So we're gonna feel thirsty, and we're going to drink water. So to bring the body back in homeostasis. So stimulus is a change in the environment. Receptors interpret that change, and then it goes to the brain or the spinal cord, and the effector is what brings about the desired change to bring the body back into the normal state. Okay, so I hope that's clear. Okay, so now I'm going to tell you how the brain is placed inside the body. So there's a skull, okay, which protects the brain, okay, and around the brain there is um, cerebrospinal fluid, so it absorbs the shock if you get hit or something. So it's attached. Um, it's it's quite strongly attached inside the skull, but it can still move. So that's why so you have to keep helmets on during sports, and I mean, if head injuries can lead to concussions and memory loss. Okay, so this is the position of the brain is very important for this chapter, because all the neurons in our body are connected to the brain and the spinal cord in some way. So it's in the skull, and then the brain stem leads into the spinal cord, and this is where the responses are decided, responses are made. Okay, so I was telling you that the neuron is like the basic unit for the nervous system. So I'm going to like go in detail into the structure of a neuron. Okay. So first of all, I'll just show you what the structures are. Okay, and then I'm going to tell you their jobs and why they're shaped the way they are. Okay. So first is the dendrites. This is where the information is received. Okay. And then we have the soma, which is the cell body. And inside the cell body, we have the nucleus with the DNA information and everything. And then we have an extension from the soma. This section, okay, where the soma turns into an axon, is the axon hillock. Okay, so this is where the plasma membrane generates electrical impulses to be sent down the axon. And as you've guessed already, axon is this wire-like structure, okay? And then we have insulation, okay? Like any wire, this wire has a insulation too. So you know how in our houses we have wires which are insulated by plastic covering. This one is insulated by myelin. So these cells are made by proteins and fatty substances. Okay, so you know fat is a very good insulator. And without this, the electrical impulses might degenerate and might be lost. Okay, and I'll tell you why there are gaps in the myelin. Okay, because you might have a question, why isn't the myelin just one big sheet covering the whole thing? why are these gaps there? So I'll answer that question in just a minute. Okay, stay with me. Okay, so this is the top part of the neuron. Okay, this is the top part of the neuron, and now I'll go to the jobs of each part of the neuron. Okay.
Could you also, Sundus, show in this diagram, you have shown the cell body and you showed axon and then the nerve ending, because I think in the book, so that they can have a holistic picture of the neuron, please. Yeah, here is it. Yep. You can exactly the same thing that Sundus has discussed. So where the impulses are received and the cell body, you could see the dendrites are there, and the cytoplasm is an extension and the myelin sheath and ending towards the nerve ending. So what is the role of myelin sheath? You will explain it here, Sundus, or later?
I'll explain it right now.
Okay, please. What is the role of myelin sheath? What happens actually if there is no myelination?
Yeah. Okay. So first of all,
so anybody would like to share what exactly would be the role of myelin sheath? Somebody would like to share?
it can be a random guess.
Keep in mind actually the transmission of impulse. Impulse is just like electrical current.
Momina?
Yes.
Myelin sheath is a fatty layer which covers the axon and it helps in the traveling of impulses.
So how does it help? I mean, what exactly is that help?
It makes the reaction faster.
Yeah, the impulse, the traveling of impulse is faster. And if there is no myelination, I think they have given an example at the same time. If you could see on the same page, 162, under this figure, there is a description from myelin, and they have mentioned if it is myelinated, so it can travel impulses 100 meter per second, 100 meter per second. But if there is no myelination, you could see 5 meter per second, so which is very slow. As you mentioned that the responses or the reaction or reflexes should be very smart and very quick. And there's one of the reason that myelin are really very, very important. Yes, go ahead, Sundus.
And then there is the disease also, multiple sclerosis, in which if the myelin breaks down, the reaction time gets less and the axon gets damaged, or the part of the body can get paralyzed because the impulses can't reach.
Yeah.
Wonderful, wonderful. Multiple sclerosis and demyelination and even neurodegeneration and also aging itself, or excessive use of alcohol, excessive use of smoking, junk food, for example, are damaging your neuron. So one of the things which is damaged is also the myelination itself. That is why people in the old age, they are very cranky. Yes, you might have heard that I cannot take it on my nerves. So what does it mean? That people get irritated. If you ask questions, if you have an argument with them, they will resort to obnoxious behavior. This is one of the things that the brains are actually, your neurons are not strong. And what will make your neuron, the more you use your neurons and the more you are get into healthy food, exercise, and that will make your neuron very, and by the way, Quran recitation, it has been proved by research that it also makes your neurons very strong. There was research going on at one student, actually, he was given a task to play guitar or music, and another student was given recitation of Quran for a couple of times. And I think one of the neuropharmacologists and neurophysiologists has done this research in our College of Medicine as well. But I'm talking about international research. So they compared actually the neuronal function after this activity for a couple of months. So the kids who are basically resorting to musical activity on and off, and the one who was reciting Quran on and off, when they compared, the neuronal structure and their function was very enriched with recitation of Quran rather than playing guitar or music. This is one research, maybe I will share with you, inshallah, the research paper, you can read it yourself. But this is a very good discussion, Momina, excellent. I would appreciate this, that you bring such a integration, with pathology or disease process. So it's not only the disease itself, genetic one, but the aging itself, age process is again very, very important. When you grow old, definitely people cannot get things. That is why memory function is affected. Their reflexes are affected. They cannot stand very well. You cannot ask a 60 years old person to have a sprint. If you compare them with a teenager or middle age, for example, there is a big, big difference. And this is one of the reasons, because of the neurons function and their myelination and other biological changes which are naturally taking place, and that will affect your physiology.
Yeah, definitely. Okay, so I'm going to first start from the dendrite, and then I will talk more about the myelin sheath. But I'm starting from the top. Okay, so the dendrites have receptors that are designed to pick up signals from other neurons. Okay, they use neurotransmitters. These are chemicals, and I'll talk more about them, okay, later on. So they use neurotransmitters for communication, and these signals that they receive cause changes in the neuron, which are then interpreted by the soma, or the nucleus in the soma. Okay, now the soma, I told you, is the cell body. It interprets the information, and it has a nucleus for DNA information, and the nucleus decides if the signal received is strong enough to be passed on to the axon helix. So what do you mean by the signal being strong enough? Have you ever noticed that if someone touches you very lightly, you don't feel them touching you at all? Even if they were sitting in front of you and touching your finger right in front of your eyes, if it's very like feather light, you're not gonna feel it. That's because the nucleus did not um, take that information as being strong enough to be passed on to the axon. Okay, so the axon helix is the junction between the soma and the axon. It's the initial segment and initial part of the axon, and this is the site where the plasma membrane generates electrical impulses to be transferred along the axon. And the axon is a long thread-like extension of the soma which conducts electrical impulses. Now this axon is covered by insulating sheath, which is myelin. Okay, so myelin is made up of fat and protein substances, and it allows quick and efficient electrical impulse travel. So how does it make it quick and efficient? This is, I mean, you will be able to better understand this if I bring the diagram in front of you, so just give me a second. Okay, so there are certain chemicals on the myelin which help the electrical transmission to, instead of flowing through the axon, it just jumps. So if you think about it, moving from, if electrical impulse was moving from the soma to the nerve ending here, and if it were to flow all the way through, and if it were to jump, it would just have to make 4 jumps and it'd be here. And if it was flowing through, it would take a longer time. Another way this is better is because all these nerves and all these neurons are bundled together in nerves. So if they're bundled together, every neuron is taking a different electrical a different information. So if they did not have some type of separation, if they did not have myelin sheath covering each and every one of them, the electrical transmission could go from one neuron and to the other. It could, short circuit, sort of.
Yes, that's very good. Yep.
So it wouldn't be like one way. So that's
Exactly.
another important job of the myelin. Okay. And if it is damaged, the impulses slow down, and you can call it insulating layer or sheath, whatever works for you. Okay, and now this is the bottom part of the neuron, the reason why I separated it from the rest is because this is actually the most confusing part. Okay, so this is the terminal, the nerve ending, right? And this is the axon terminal. People also call it the synaptic button. When the signal reaches the axon, these terminals release a chemical called neurotransmitter. If you recall it, this came up before also, and now I'm going to explain what a neurotransmitter is. So first of all, if I go back here, these parts are actually receiving information from yet another neuron. So this is receiving information from another neuron, and
So what do you call that information in terms of these book? They have mentioned what exactly for this information? Stimulus, right?
yeah.
Yeah, this is the stimulus. When stimulus is received there, okay, and then it will generate a current through the through the axon. Okay, go ahead.
So what I was trying to say is that this if you look over here, this is the ending of one neuron and this is the beginning of the other neuron. So this is the axon terminal of one neuron and this is the dendrite of another neuron. So I want you to really understand this before we go on. Okay, so this is not singular synapse. This is a mistake. So one of these junctions is a synapse and these are synapses. Okay, now if I zoom in on one of the synapses, it looks something like this. Okay, so this is the prosynaptic neuron and this is the postsynaptic neuron. So presynaptic neuron, you can judge by the name. It comes before the synapse, and this is after the synapse. Okay. So let me just discuss the structure, and I'll tell how this works. Okay. So this is the axon terminal. This is the dendrite of the next neuron, the postsynaptic neuron. There is space between these this axon terminal and this dendrite. It's called the synaptic cleft. Okay, and so this whole thing is this area. This is what is a synapse. Okay, and these are receptors. Okay, and now I'll tell you what the neurotransmitters are. So the neurotransmitters are chemicals in every axon terminal in the presynaptic neuron. Okay, so this is the axon terminal. These vesicles contain the neurotransmitters, and the neurotransmitters contain the information, the stimulus, um, over here. Okay, then this at the at this at the axon terminal, the vesicle moves when the electrical impulse comes. Okay, so let me begin again. So if when the electrical impulse comes in, okay, so the neuron becomes kind of activated or excited. Okay, and that's when the vesicles move closer to the cell membrane and it ruptures. When it ruptures, the neurotransmitters are released into the synaptic cleft, and then when they diffuse to the receptors, and then the receptors. So these are gateways. Okay, so when these neurotransmitters attach themselves to the gateways, these gates open up. When these gates open up, that's how the electrical transmission can then go to the postsynaptic neuron, to the dendrite. And this is how it's just going to continue on. It's like a wire. It's going to go to the brain or the spinal cord. Okay. Now, the once this and the electrical transmission is done, what is going to happen to the neurotransmitters which are left here, because they were just there to open these gates, right? So either they can be taken up by this voltage gate. Okay, So why this I'm calling this the voltage gate is because this actually opens up only when there is some potential difference. So this is where physics comes in. So if you remember, potential difference was the separation of charge. If you've done circuits, it's the separation of charge, separation of positive and negative charges, right? So when there's a voltage, so a voltage is a potential difference, remember? So when there's a voltage, there's a potential difference, and that opens up this gate. When this gate opens up, the neurotransmitters are taken in and back into the vesicles, okay?
Yeah, would you like me to add a little on this here? This is
Yeah,
really very
sure.
nice explanation. Yeah, as you mentioned that the vesicles, when they are after arrival of the signal at the axonal end, so the vesicles are basically releasing neurotransmitters in the synaptic cleft. So these neurotransmitters are really different for different organ system. Like for example, in the heart, if you want to increase it, there could be adrenaline or also acetylcholine, for example. They are different chemicals actually. They're not always the same. That is why your organ responds differently. One thing. And another thing, at the cleft, there are certain enzymes. As you know that there are many millions of transmitters are released. Actually, you need few of them because you could see here in the postsynaptic neurons, you have receptors here. So there are these transmitters actually is like a key and lock model. So these transmitters are easily recognized by these receptors, and they attach to the receptor is stimulated, and this signal is propagated a little further towards the organ or through the whole transmission system. So whatever transmitters are left behind, there are two facts for there are two consequences, there are two mechanisms. One thing is metabolism, so these are degraded, okay, but majority of the transmitters are preserved. They are taken up back, as she mentioned, the gates, and these gates are called reuptake transporters. These are reuptake transporters. Reuptake means they are taken up back. So they are taken up back by these because of the voltage difference. They are open. So this is one of the mechanisms that your body should not lose. Otherwise, if all the transmitters are metabolized, so what will happen? Your body will spend more energy and more efforts to synthesize them and to refill these vesicles. So this is a beautiful system. That is why they are taken up back by these transporters, and we call it reuptake pumps. We also call it reuptake transporters. In people who are suffering from depression, we are using actually drugs that block these transporters, so that, because in case of depression or anxiety and other psychological states, especially in major depression, where you feel very lonely, sad, inactive, and doesn't take much interest in life. So these cleft have very low level of transmitters. So if they are released and they are taken up back by these transporters, you have very less neurotransmitters to be available in the synaptic cleft. So how you can increase these transmitters in the synaptic cleft? That you block the transporters, reuptake transporter by drugs. This is only for those patients who are suffering from depression, okay? For normally, you don't need it. Normally, this should be very active. Otherwise, patient will go into mania or psychosis. They will respond hallucinations, and very bizarre behavior, for example, very aggressive, insomnia, lack of sleep. So I just wanted to integrate this. This is a very nice picture that you will see also in the medical books. When we talk about antidepressant drugs, people, inshallah, when you go into medical schools, you definitely would rely on these diagrams in these figures. Excellent, thank you.
What the degradation process, as you mentioned, is done by specific enzymes which break down these neurotransmitters into their basic components, which all this neuron again then takes back in and makes new neurotransmitters. Okay, so a synapse, to sum it all up, a synapse is a place where two neurons come so close together they can pass chemical signals. Okay.
So synapse is made of what? So if in a little fine details, how you will define synapse itself?
A synapse is
this whole structure that you are showing, the combination of presynaptic Postsynaptic in the space itself.
Yeah.
This whole area is called synapse. So
This
sometimes
whole,
people are confusing synaptic cleft with synapse. Synaptic cleft is just the space. It's not synapse. Synapse is this combination, the whole, the space, the two neurons, pre and postsynaptic. This is called synapse, which is also described in your book.
yeah.
Okay, go ahead.
This is a part of a synapse, and this whole thing, these are the components of the synapse. Okay.
That's cool.
So okay, so a synapse, you can say, is a connection between neurons. But this definition is also wrong in the fact that neurons can also be attached to the effector cells. Remember, the effector cells are the cells or the organs which bring about a change to bring the body back into the normal state, remember? So they can also be attached to the effector cells, so that to affect the effector cells, obviously to activate the effector cells. So that is also a synapse, okay? So I'm just gonna mention a brief history over here. So there was a scientist named Otto Lowy, okay, and he had, I don't know how true the story is, okay, so he had a dream in which he saw an experiment. So when he woke, he wrote the dream down, but then when he woke up, he couldn't read his handwriting. But thankfully, he had the same dream again, and this time he did manage to read his handwriting. And what the dream said, what the dream was, it was an experiment on the heart of a frog. Okay, so in on one frog there was the nerve was intact. So when there was an electrical impulse through the nerve, the heartbeat slowed down. And then he took another heart from another frog, but this time he removed this nerve, okay? And he got the heart moving. So the heart was pumping, but but there was no there was no nerve. So then using a pipette, he took the fluid from this heart and then transferred it to this heart, and this heart slowed down despite the fact that it didn't have a nerve. So he came to the conclusion that it must be something with the fluid, not the nerve. And that's how the neurotransmitters were discovered. So now we know that even though the nerves and the electrical impulses are a big part of the response, it's these neurotransmitters are way more crucial. These are the chemical substances which bring about the change. Okay, so now there are two types of synapses. One is a chemical synapse and one is a electrical synapse. So a chemical synapse has a greater synaptic cliff, a larger synaptic cliff, but electrical synapse does have a synaptic cleft, but it's smaller, okay? So now in a chemical synapse, as the name suggests, we use chemicals. Okay, so again, over here, these are the vesicles, and this vesicle bursted, ruptured, and these are the neurotransmitters, these are the gateways, the receptors, and this is a dendrite, this is the axon terminal, and there is an action potential. Again, the term potential. So this is just like circuitry in physics. So potential, again, difference in charges. So I'll tell you what action potential is when we move on. Okay, so the action potential, it activated these vesicles. The receptors uh, received the neurotransmitters. The gateways were open, and uh, uh, the electrical thing was led to transmit. Okay. Then in the electrical synapse, there are voltage gated channels. Okay, so these channels open up just by the difference in potential, okay, the voltage. So whenever they feel the voltage in the neuron, these channels are open, and so the electricity passes through. Okay, not electricity, electrical impulses, but yeah, you can think of it as electricity. Okay, so these are two types of synapses. Would be chemical synapses are more common because electrical synapses are not controlled. Okay, and so there is danger of like short circuiting. This is used in few parts of the body, like the retina, where the response needs to be very quick. This is used in controlled like muscles and stuff. So whenever you want to stop, you can stop. Okay, and whenever you want to start, you can start. Um, so now comes the question: what is a action potential? So there are three types of potentials. One is the resting potential. Okay, so this is just a name because neurons are never resting. Your brain never rests because your body depends on your brain. Even when you're sleeping, you do need to breathe, and your breathing is controlled by your brain. So this is just a name. Okay, so there's a resting potential, graded potential, which is less common, and then the action potential, which is very crucial. So the resting potential. So I'll talk about the resting potential first. So first of all, potential is just a separation of charge. So if this was the neuron, the axon, there would be negative charges inside and positive outside. And in a resting neuron, so when you're not doing anything, okay, but you are going to be breathing and stuff, that's how you're alive. But you know, technically, if you're not doing anything, the voltage would be minus 70 millivolts. That is the resting potential. Okay, so that's the resting separation of charge. Okay.
Yeah, so the way, would you like me to add here
Yeah,
something
yeah, sure,
regarding
sure.
the resting potential is also called baseline potential. Actually,
Yeah,
this is highly needed. The electrical impulse is not a continuous process. What happens basically, the impulse is transmitted by the flow of ions, sodium and potassium, right? And
yeah,
this is how it generates impulse. So what happens basically, it changes the potential inside and outside membrane, and this is how the impulse is transmitted. So initially what happens actually, sodium channels are open and there's influx of sodium inside the cell and outflux of potassium. So when this impulse is traveled from that area is going to another part stimulated. So the part where the action potential was existing, that will come to the normal stage. This is called or baseline potential, or we call it resting potential. It's exactly not resting, but it is now able to receive signal, another signal. It's now
yeah,
very able, because if you change this, the potential, for example, in case of diseases, so your muscle will not respond. That's why some kind of potential of resting threshold is needed. So if there's little change, so you all of a sudden the electric impulse will be generated, and this is why we call it resting potential. So we call it depolarization and repolarization. Depolarization means now the membrane is stimulated, and then repolarization is coming back to the normal stage when there was no impulse at all. It's not going to participate in the impulse transmission because the impulse is actually past that area, and now that area is now able to receive new impulse. That's why we call it resting potential or baseline potential, and this is something that you need to understand.
yeah. And I will clear it up as we move on. So over here is a basic graph. So on the y-axis we have the membrane potential, and over here on the x-axis I have time. So you can see that the resting potential is minus 70 millivolts. Okay, I forgot to write the units. Never forget to write the units in the graph, okay? So this minus 70 millivolts, okay? And then you can see that it's rising. It's rising, right? So this point, okay, so let me make it more clear. Um, so this point over here, this is the threshold. When it reaches this threshold, this is the point when all those voltage gates and all those neurotransmitters get activated. And then you have this activation of potential. And once the electrical transmission is transmitted, comes back to normal, and you have this dip, and then it comes back to -70. So all of these stages have names, which I'm going to discuss as we move on, but I don't want to confuse you right now. Okay? So now I want you to picture what happens at the, the synapse, okay, as the electrical impulses reach it. So if this is the membrane of the synapse, okay, and I like to think of this to remember all these ions. I like to think of a salty banana. So a banana, we all know, has a lot of potassium. So thinking that this is a banana, I will make a lot of potassium over here, and there is a bit of potassium outside, but not too much, okay? And now because it's salty, it has sodium and chlorine outside the membrane. A bit of sodium and chlorine inside too, but it's not too much. So you can see the gradient, right? This is an ion, it's a protein, okay? And so this is how the ions are set in the resting phase, okay? Now when the electrical impulse comes, okay, so basically, before I get into it, uh, let me tell you one more thing. There, in these membranes, there is a leak channel, okay? And pay attention to the word leak, okay? So it allows the ions to go both outside and inside, okay? So there is no one-way flow here. And leak also means it's small, so these big proteins cannot go through. I'm going to show you a simulation of this too, okay, but for now just bear with my diagram, okay? So you can see that there is a high amount of potassium here and less amount of potassium here. I've just drawn one, okay, but there are a couple more potassiums too, but they're less than number over here. And I've removed the sodium and chloride from both sides, okay? Now you can, if I drew a graph of the potassium ions over here, and if my y-axis was amount of substance and the x-axis was time, I will see that there will be a flow of as time passes by, there will be a flow of potassium from the high potential to the low potential down the concentration gradient. It's going to diffuse out, right? But as it starts diffusing out, as it keeps diffusing out, down the concentration gradient, right? But as it's diffusing, so just to give you a clear picture, let me draw. So it's diffusing out, okay? So there's more potassium here, and so these ones are gone, okay? You can see that the gradient is decreasing. Now if you count the number of potassiums, I have 4 potassiums here and 4 potassiums here, so there is no gradient, right? So now if a potassium flows in here, this area is going to have a positive charge, and this area is going to have a negative charge, overall negative charge. I know this is a bit confusing because there are all these potassium ions over here, and there are cation ions, and there's a negative charge here, but
Yes.
this has a positive charge outside the membrane, and inside is a negative charge. So if you have any question right now, please ask me because this is really important.
Yeah, and also Sundus, yeah, you give them time if they want to ask some questions. So Sundus is basically explaining here the impulse transmission. Actually, this is the mechanism of impulse transmission through the neuron, okay? So this is how the impulse is transmitted from one neuron to another, and how they are releasing these transmitters in the synaptic cleft. So go ahead if you have any question, any query, any clarification you want.
Before I move on. Okay.
You have any?
There's no question, I think. Okay. So I hope you understood that, okay? So there's negative charge here and positive charge here, relative negative charge here, relative positive charge here. We know that we do have chlorine ions over here, sodium ions here too, okay? But there's a relative negative charge here, relative positive charge here, okay? So now, as this happens, we notice a electrical gradient. So using the convention, the electrical gradient is from the positive to the negative, though I know the electrons move, but we're using the convention. Remember in physics, the conventional flow, right? So I have made the arrow from positive to negative, even though the electrons will be the one moving, okay? So um, relative positive charge here, relative negative charge here. That's how the electrical gradient is made. So why is the electrical gradient important? Okay. Uh, so I'll come to that in just a second. But first of all, let's come back to the action potential. Okay. So you saw what was the potential, right? It was the difference in the electrical charges. The electrical gradient is the potential. Separation of charges is the potential. And now the action potential is when these voltages actually open these gates. So let me label these gates for you. This is a sodium gate. So this is a sodium gate, and this is a potassium gate. Okay, and now I also want to keep the graph in front of you. So if you look at the graph, minus 70 was the resting potential, right? I was talking about the threshold. So it was increasing. Okay, the potential was increasing, and when it reached minus 55, it activated. So this is the potential in millivolts. Okay, so when it reached minus 55 millivolts, this gate got activated and it opens up, and it opens up really wide, so a lot of sodium comes inside into the synapse, okay? And that's what causes this sudden peak, because you have that sudden influx of ions and then a very fat, I told you, the movement of ions is what makes the electrical gradient. So if you have a faster flow of ions, you have a faster increase in the electrical gradient. So and now it comes into the positive. So it comes into the positive 30. So now when it reaches positive 30 millivolts, it activates this gate, which is the potassium gate, right? So now and now remember, this sodium is a cation, okay? So there was a influx of cations. So let me just write that down. There's an influx of cations, and now there is removal of cations. So this is going to like sort of cancel it out, right? So when it reaches positive 30 millivolts, this gate gets activated. This gate opens up and potassium starts leaving that cell. And so we have polarization, we have the canceling out of the charges, and it goes down, the potential goes down. And then a time comes when this gate shuts. So this gate shuts, but this is still removing potassium. So this goes down. It's called a undershoot. It goes below normal. But then both of these gates regulate, and this one shuts down too. So does this, and it comes back to minus 70 millivolts. I know.
Yeah, and you could Sundus also mention about this depolarization is the process when these gates are activated, and the impulse is basically transmitting in that time, and then membrane is basically activated for the channel for the ion exchange. And repolarization, you could see here, is bringing back to the resting phase. Re for resting, bringing back to the resting stage.
Yeah, exactly. So
Okay.
I hope you understood this. I mean, if there's any question, I know this is really complicated. So I want you guys to ask that question right now.
This is really a very nice concept. Actually, we call it electrical physiology. This is electrical physiology, and you would study that, I think, a little higher level, though it's not mentioned in the book. But thank you, Sundus, for the great effort that you clarified it very well. We basically teach this in our medical students' first and second year physiology. Those who are physiologists, they definitely, that maybe those are medical doctors who are teaching. They must be very much aware with this concept, which is really very relevant. So this is the whole process of transmission. And keep in mind, when you touch a hard surface, so this is what's going on, before you contract or before you withdraw your hand. So these all processes are going on. All of a sudden, in a split of second, you just touch the hand, touch the hard surface, and all of a sudden, these ion exchange takes place. The impulse is generated, and that is how sensory neurons is communicating that to the brain, to the spinal cord, and spinal cord is executing basically through the motor neurons, bringing back these signals through these ion exchange, these ion systems. So when these ion are exchanging, they are basically transmitting the impulse. When it reaches the effector, so effector means these nerves are basically now releasing some chemical, these neurotransmitters, to the organ. In this case, the reflex arc, your arm muscles, basically. The muscles are contracted, and this is how you withdraw your hand immediately. So all these ion channels in split of second. Subhanallah, this is again a beautiful creation of Allah subhanahu wa ta'ala. What a great mechanistic, what a great harmony. And even a single, split of second, you could say there is no deficit or any fault in the system. Otherwise, it will ruin the whole way, the whole process. So, alhamdulillah, it's very well coordinated.
Yeah, this happens in just a millisecond. So now this is where I said that I'll answer why the myelin sheath needs to have gaps, right?
We need to wind up in 5 minutes, Sundus?
Yeah, sure.
Okay. So because
so the reason why this myelin sheath needs to have gaps is because voltage gates need to be having a connection to the external environment of the neuron, and they can't really be releasing all these ions into the myelin sheath, right? So they need this gap. This is where the voltage gates are. So now if I zoom in, this is what happens. So this is the myelin sheath, okay? Here are the voltage gates, okay? Here are the voltage gates, okay? So there are many voltage gates, but I'm just talking about the potassium and sodium ones, okay? So here is what there's going to be a positive charge. Over here, there's going to be a negative charge, okay? Because there are no gates here. There's nothing to balance out that electrical gradient, okay? So this is how the electrical impulse jumps. Can you see this is positive in between? This is negative. So the electrons jump from the myelin sheath on to this section, and this is how the electrical impulse moves along really fast, and it's interchanging all the time, okay? So this is really complicated, but I hope you did get a rough idea of what happens. If you have any questions, you're free to ask. Okay, so if there are no questions, let me just show you the simulation. Okay, let me just share my screen. This will be the last thing. Can you guys see my screen?
Just wait, I think it's going to be started sharing screen. Yeah, yes.
Can you see it?
Yeah, we can see it, yes.
Okay, so here I'm zooming in to, okay, so here I'm zooming in to the membrane, okay? So let me zoom in, okay? And I'm gonna put it on slow motion. I'm gonna stimulate the neuron, which means that I'm gonna make a electrical impulse pass through. So you're gonna see a influx of ions. So here's the, can you see there are so many ions? So this is sodium coming in, the diamonds are like the potassium, and now the gate's shut again. So if I showed you the chart, you can see it again. Just give me a second. So I'm gonna put it on slow motion. Okay. So we stimulate the neuron, and if you look at the chart, can you see that it's rising? And now it comes back down when the potassium goes out. So this is what I wanted to show you because I couldn't in the diagram. Okay, so I hope it's clear.
Yes.
And if there's any question, you guys are free to ask.
They can raise your hand, and then we'll take just 2 minutes to show the book on page, I think, 162.
Okay.
Yeah, and there are few things they have discussed here, coordination in animals. So I think this is just only a few things like what is stimulus. First of all, the core knowledge is what is stimulus. Stimulus is the one that is received by your sensory neurons. For example, if there is drop in temperature, you will see your body start shivering, right? And you would see that your skin is very much pale or constricted. The vessels are very constricted. While in case of hot summer, you would see that the skin vessels are dilated and body will start sweating because this change in temperature is the stimulus. So that is actually sensed by the receptors in the skin, right? That is how these receptors, so stimulus is received by the receptor. And then the receptors are basically propagating all these transmission through the sensory neurons, and there are different kinds of neurons. Inshallah, in the next lecture we'll talk about. And then you have motor neurons that are bringing, you know, for example, in case of reflex arc, this will cause reflex action. You will actually immediately withdraw your hand if you're touching a hard surface, or if there is drop in temperature, your blood vessels are very much constricted. That's why in hot summer you would see in the skin it's very easy to draw blood, or because you know the vessels are very much dilated, because you want more blood flow so that the body heat could be, your body is basically protecting you against the overheating. This is a natural process. You have in the car engine, you have fans there, but also Allah subhanahu wa ta'ala has created a system when there is too much hot, a high temperature outside. So that is sensed by your skin, processed by your central nervous system. And then central nervous system has different mechanism, threshold for the temperature in your body. It has to maintain a normal temperature, which is needed for physiological function. So it does not lead your body to be overheated, because that it will damage enzyme system in your physiological process, and many mechanisms will be damaged. This is why the body has to restore that temperature. So these are all controlled by central nervous system through neurons. You know, these neuron system, you have stimulus, you have receptor, you have effector. Effectors are the organ itself. Like for example, there could be gland also. Even the glands are under the control of central nervous system, right? They are really like growth hormone. Some things are really needed on gradual basis. Like for example, growth of body is a slow process. So this is why pituitary gland is under the control of hypothalamus, the central nervous system, releasing growth hormones, and they are working very gradually, working on the bones, working on the muscle, working on the shape of the body. You know, all these are gradually changing. While in case of heart rate, blood pressure, body temperature, urination, all these need very fast action. And they are certainly under the control of hormone, as well as under the control of the nerves. The neurons itself are controlling your body function, changing in the blood pressure, changing the heart rate. Like if immediately you start climbing stairs, you are going upstairs, so you are basically, your heart should have more pumping ability. That's why it will pump more blood. And how it can do that, the moment you step up, you know, going upstairs, so this is actually sensed by your body system, the pressure itself, and it send the signal to the central nervous system, and then central nervous system will release different hormones in the body to increase the heart rate, to constrict the vessels, for example, so that blood can go very fast. The respiratory rate is increased, oxygenation of the blood is very quick, carbon dioxide, which is generated very quickly in the body, you are getting rid of that is very, very quickly. So these things are very well coordinated.
And vasodilation is also the reason why nosebleeds are more common in the summer, right?
Yeah, this is one thing. Very nice. Yes, yes, very true. People, yeah, because these small capillaries, especially the vessels, are dilated, so you would see more blood flow. And this is why you give them vasoconstrictor. The nasal drop that you are taking for, this is called epistaxis, so the nasal bleed, to control that, you basically give a chemical, which is vasoconstrictor. So it constricts the vessel. So basically that blood flow is decreased to that particular area. And this is how your dentist, when you go for dental procedure, they will give you vasoconstrictor as well. Because if they extract the tooth, so what will happen? You probably expect more bleeding, which is dangerous. So the doctor would give anesthetic, local anesthetic, and then also vasoconstrictor, because in that area, this is a rich area for blood flow. So you want to constrict the vessel so that you can decrease the blood flow. I mean, this is how it will control the bleeding as well. Yeah, it's a very nice correlation. Today, actually, we went a little bit, I would say, in a very nice foundation, good foundation for the whole chapter. And then don't worry, we will cover all these topics in a sequence. But I think this lecture was really a foundation of central nervous system physiology, I would say. If you are talking about the coordination system, so you go through these, the description in the chapter on page 162 and 163, and inshallah in the next lecture we'll talk about it, what exactly is coordination, what is stimulus, and the whole process, you know, is called coordination. Coordination means you are making use of the system, the stimulus, how it is received, and how it is propagated or transmitted to the brain, and how the brain is controlling, reacting to that particular situation. For example, in case of touching a hard surface, your muscles are contracted and you withdraw your hand. So this whole process is called coordination. So this is coordination, and the withdrawal of your hand is the response. This is the reaction or response that you are basically responding. And you would say, as mentioned by Momin earlier, in some diseases, for example, in case of paralysis, polio, or that part of the body or organ is basically not under the control of CNS. There is a problem with the neurodegeneration, or nerves are damaged. So even in that case, if somebody touches the hard surface, the whole, hand will be burned, but patient won't feel it. So this is very, very important. People who are taking narcotics, this is why you don't leave them under near the heater, or don't leave them driving, because they cannot have efficient response system. So if they touch any hard surface, their threshold is very high, you know. Sorry, their threshold is very low, right? They will respond very slowly until the skin is damaged, and if there's a huge damage is done, only then they could respond, because they are under the anesthesia. For example, when patient will go to anesthesia surgical procedure, if they want to remove the kidney transplant, if they want to replace the knee, or they are going to do any other surgical procedure like open heart surgery, for example. So the body should function, but your patient should not respond. The pain receptors are suppressed. Your higher center is suppressed, is not responding to the stimuli. This is why patient cannot move until the procedure is done. So if you want to have any comment, I can give you 1 minute, and then we will conclude the meeting. And inshallah, see you tomorrow at the same time, 1:45 p.m. Is there any comment, anything you would like to share?
Any question?
Yeah. So I think tomorrow we're going to explain reflex arcs.
Yeah, tomorrow I'm going. So this was like the basic, because I wanted you to know about the basic unit of the nervous system. And then tomorrow I'm going to talk about the nerves and how they're arranged, and the transmission of electrical impulses. So,
All right, excellent. That will be great.
okay. So I don't
Okay,
think you
inshallah.
have any question.
I don't think, yeah. So see you all inshallah tomorrow, same time. Jazakallah khairan. Have a nice day.
Allah Hafiz.
Allah Hafiz.